UN 영어뉴스. First synthetic human embryo raises ethical issues. Scientists have created the first synthetic human embryos using no eggs or sperm, provoking deep ethical questions, according to reports. The synthetic embryos, only days or weeks old, could help researchers study the earliest stages of human development and explain pregnancy loss. Nobody is currently suggesting growing them into a baby, but the rapid progress has outpaced discussions on how they should be dealt with ethically and legally. Prof. James Briscoe from the Francis Crick Institute said the field needed to proceed cautiously, carefully, and transparently to avoid a chilling effect on the public. The development of human synthetic embryos was announced at the annual meeting of the International Society for Stem Cell Research. Synthetic embryos are also known as embryo models, as they resemble embryos for the purposes of research rather than being identical to them. The work comes from the laboratories of Prof. Magdalena Jernicka Goetz from the University of Cambridge and the California Institute of Technology. The full details have yet to be published and made available for scientific scrutiny, leading many researchers to feel unable to comment on the significance of the reports. But the principle is the synthetic embryos are made from a stem cell rather than a fusion of egg and sperm. Beating heart, stem cells have the capacity to become any cell type in the body, and if coaxed in just the right way, can be persuaded to form embryos. And this is the first time that has been achieved using human material, although they are not truly synthetic, as the starting material was cells cultured from a traditional embryo in the laboratory. It's beautiful and created entirely from embryonic stem cells, Prof. Jernicka Goetz told the Guardian newspaper. She has already developed synthetic mouse embryos with evidence of a developing brain and beating heart. Meanwhile, scientists in China have implanted synthetic monkey embryos into female monkeys, although all the pregnancies failed. Side by side, the natural and synthetic mouse embryos looked very similar after 8 days. The synthetic embryos do not behave in exactly the same way as normal embryos, and it is unclear how their use in research should be governed. Prof. Briscoe said, on the one hand, models of human embryos made of stem cells might offer an ethical and more readily available alternative to the use of IVF-derived in vitro fertilization human embryos. On the other hand, the closer stem cell-derived models of human embryos mirror human embryos, the more important it is to have clear regulations and guidelines for how they are used. Most countries use the 14-day rule in human embryo research. This allows an embryo created by fertilizing a human egg to be grown for 14 days. However, these embryo models are not legally embryos and are not governed by the same laws. Dr. Ildem Ackerman from the University of Birmingham said, "These findings suggest that we would soon develop the technology to grow these cells beyond the 14-day limit, with potentially more insights to gain into human development. Nevertheless, the ability to do something does not justify doing it. Understand infertility. Legal and ethical experts in the UK are drawing up a voluntary set of guidelines for how to proceed. Researchers hope these synthetic embryos will further understanding of the earliest stages of humans' lives." Prof. Roger Sturmy from the University of Manchester said, "We know remarkably little about this step in human development, but it is a time where many pregnancies are lost. So models that can enable us to study this period are urgently needed to help to understand infertility and early pregnancy loss."